Hi class, this is Mr. Durst, and in this chapter we're going to be focusing on the physiology, the chemistry of the skeletal muscles in our bodies. This chapter is kind of beefy. There's a lot of content in this chapter, and we're going to introduce electrical activity of cells, and we're also going to focus on how we can take the big muscle itself and make that whole muscle move from an individual molecular level with chemical basis. So this presentation, this chapter has been broken up into multiple presentations. This is a beefy chapter. I want to encourage you to read the textbook chapter in its entirety. And this is the very first time I'm ever going to tell you to read the textbook chapter in its entirety because this is a really difficult chapter to go through. So let's get started. As we look at muscles, there's lots of characteristics of muscles. One of the big ones that I emphasized when we were talking about the histology of tissues of the body is that skeletal muscles or muscles in general can contract. And this is a trait which is unique to only muscular tissue. Only muscular tissues contract when we stimulate them. But there are some other characteristics that all muscular tissues have. For example, they can transmit electrical signals across the surface of their cell membrane, known as sarcolemma. They're also going to be able to respond, or they're excitable. So we can have chemical signals, physical signals, electrical signals, causing some kind of a response or an alteration to that muscle tissue. They are also extensible. What does extensible mean? It means that we can stretch them out. When we're relaxing our skeletal muscles in particular, we can stretch them out beyond their resting length. So if you look at a muscle at its regular resting length, you can pull it farther. This is a key characteristic or a key way that fishermen or fisher ladies, anybody who loves to go fishing, will take their fish and they'll kind of hit it on the edge of the boat and then stretch it out to maybe take that fish that's not quite long enough for the legal limit and stretch it out an extra half an inch to make it long enough so that they can keep it as opposed to throwing it back. However, there's a problem. After stretching out a muscle, or in this case, stretching out your fish so it's long enough for the legal limit, if you let go, a lot of times those skeletal muscles, or the muscles in general, will swing back to the original resting length. And that's referred to as elasticity. Extensibility and elasticity are oftentimes mixed up by students. So when we think of extents, think of making the muscle longer. When we think of elasticity, think of returning or springing back like a rubber band to the original resting length. Now, we're going to spend most of the time in this chapter focusing on skeletal muscle tissue because most of the muscular tissue in our body is skeletal muscle tissue. So as we look at our skeletal muscle tissue, it's voluntary. We can choose when to contract it and choose when to relax it. Also, as the name skeletal implies, it's almost always going to be attached to bones in our bodies. Every once in a while, there's a little bit of an exception. The general rule of thumb is that skeletal muscles are attached to bones. A key characteristic of a skeletal muscle is that it has highly developed, very pronounced striations. I'm drawing some arrows. I've circled this term. Skeletal muscles have the biggest, boldest, baddest, most notable striations of any of the muscle tissue types. And when we think of these striations, a good synonym is going to be stripes or light, dark bands that go across the cell short way or the transverse distance. And these striations can be thought of as a way to visualize contractile proteins within a muscle. So the more striations you see, the more force that muscle cell or that muscle fiber can contract with. Now, what do I mean by voluntary? It means conscious control and usually subject to conscious control. There's a couple obvious exceptions. For instance, there's reflexive responses. If somebody tickles you, a lot of times you start twitching your skeletal muscles involuntarily. But most of the time, you're going to choose to voluntarily contract those skeletal muscles. Or the other big exception to this rule is the diaphragm. By default, our diaphragm will contract and relax rhythmically to help us inhale and exhale, to move air in and out of our lungs. But we do have the ability to override the involuntary contraction and relaxation of the diaphragm to hold our breath or to force air out of our lungs with extra force. Sometimes that happens, though, involuntarily when you cough. And no, I didn't do that on purpose. It was just great timing. Now, as we look at a muscle fiber, It's a synonym for a muscle cell or a myofiber. Myo meaning muscle. And the reason they're called fibers is because they're super skinny. They're about 30 to 40 microns across in the short distance, but they're 30 centimeters long, and they can go from the thigh or from the hip down to the knee. And these muscle cells are so long and filamentous that oftentimes they'll just be referred to as muscle fibers. So here's a photomicrograph of some skeletal muscle fibers. Note we can see how there's some striations, there's some bending across that transverse distance. And because our skeletal muscle fibers are so long, they are going to be multinucleate. This one nucleus can control or send signals that control one area of the skeletal muscle fiber and its proximity. And then an adjacent nucleus, we'll say over here, will control this area of the skeletal muscle fiber. So these multiple nuclei are going to control the region, their local neighborhood of the cytoplasm. And this is one of the ways that we can have a cell be 30 centimeters long, but still account for the diffusion of the signals from the nuclei, so we can have chemical reactions take place in a timely fashion to prevent cell death. Now, we had mentioned these briefly in our Chapter 10 presentation. I'm going to cover them very quickly right here. There are connective tissue wrappings around our skeletal muscles. We have the epimysium on the surface that holds all the fascicles together. Individual fascicles are going to be bundles of muscle fibers held together by a perimysium. And within fascicle, we're going to have individual muscle cells, individual muscle fibers being electrically insulated from the adjacent muscle cell with the endomysium. Tendons are made of a lot of collagen, and they're going to attach a muscle to a bone. And then as that muscle, the collagen fibers go from tendon to the bone, they're then going to embed themselves within the extracellular matrix of the bone. This collagen that makes up the tendons and a lot of these connective tissue wrappings is very tough. It's extensible and elastic. What does this mean? Our collagen can bend. It can, instead of fracturing, it flexes. Instead of breaking, it bends. So because it has just a little bit of flexibility, collagen is very resistant to stretching forces and does a great job of resisting tears from pulling forces. And this collagen is what's responsible for the extensibility and the elasticity of muscles. It's also worth emphasizing that this elastic recoil of the collagen fibers gives us a little oomph or a little bit of extra power to our steps that allows us to increase the power output of a skeletal muscle by just a little tiny bit. Now, within one individual skeletal muscle fiber, aka a skeletal muscle cell or a myofibril, we're going to have a fancy name for the plasma membrane, the sarcolemma. And here's the third word or prefix that I'm going to introduce in this class for muscle. We've had myo, mys, and now sarco. Sarco means muscle. You've probably heard of sarcasm or sarcastic. If you look at the original Greek root words, to be sarcastic is to tear flesh from bones. So that's one of the reasons why sarcastic people oftentimes hurt other people's feelings. They're making these tearing or these put downs, these belittling comments. Back to A&P. Sorry about that tangent there. So sarco means muscle. Sarcolemma is the plasma membrane or cell membrane of muscle fiber. And instead of just saying the cytoplasm, we're going to say the sarcoplasm, the cytoplasm of a skeletal muscle cell. Now, 2 presentations ago, I mentioned how skeletal muscles are bundles of bundles of bundles. And I mentioned how we have an epimysium bundling the fascicles, and within the fascicle, we have muscle fibers bundled together. Within muscle fibers, we have more bundles. And these are going to be myofibrils that are bundled together in a muscle fiber. We're also going to store a lot of complex carbs known as glycogen so that our muscles have access to carbohydrates to turn into energy, ATP, and we're also going to store oxygen on individual myoglobins, globular proteins unique to muscles. And there are reg pigment that have an iron atom at the core. One of the key differences between those of you who like to eat fried chicken, one of the key differences between white meat and dark meat on the chicken is going to be myoglobin concentration. Muscles of the legs are constantly working, so those muscles of the leg on a chicken are going to have more myoglobin. Those will be the dark meat. Chickens don't fly very well. Their breasts or the pectoralis major muscles on a chicken don't do that much contracting, so they don't need a lot of myoglobin. So consequently, chicken breasts are white meat instead of dark meat. Now, within our skeletal muscle fibers, they are multinuclei or have multiple nuclei. And there's a different ways, schools of thoughts of why this is going to happen. We take an origin school of thought. We look at the myoblast, the muscle building cells. Blast for build, myo for muscle. A muscle builder is a myoblast. These are multipotent or adult stem cells that fuse together end to end to form a very long skeletal muscle fiber. We're also going to have satellite cells, and these satellite cells are unspecialized myoblasts that are going to remain within the endomysium, and this is going to give us a small ability to regenerate skeletal muscle tissue. As we age, we rapidly lose these satellite cells. Young children that have major damage to their skeletal muscle tissue can oftentimes regenerate and recover from that very quickly. Because our skeletal muscle fibers have to burn through so much energy, they're going to have lots of mitochondria within them to generate lots of ATP to be used as a source of contractile energy. There's also, within our skeletal muscle fibers, some unique things, such as a variation of the endoplasmic reticulum known as the sarcoplasmic reticulum. And the sarcoplasmic reticulum is going to be primarily a way to store and release calcium as we modulate or regulate the contractions of these muscles. And within the sarcoplasmic reticulum, we have some bulges that are at the endpoints, referred to as the terminal cisterns. Those terminal cisterns are going to be on this figure as the blue tubes. And then this blue mesh, or this blue network or spider web, is the rest of the branches of the sarcoplasmic reticulum. In between the terminal cisterns of the sarcoplasmic reticulum, we have transverse, and you should write this in the margin, T stands for transverse tubules. And these transverse tubules are going to be invaginations or infoldings of the sarcolemma. So here we can see some little tiny dots represent openings and within the cell membranes, or holes in the cell membrane. And those holes are going to form this purple tube that goes all the way across the muscle fiber to the other side. That's the transverse tubule. It transverses the muscle cell from one side to the other side, short direction. And if we take one transverse tubule and pair it with the two terminal cisterns, we form a triad. And this is important because as electrical signal travels across the skeletal muscle cell or across the sarcolemma, that electrical signal, let me clear the screen here. So the signal goes across the surface. Electrical charge or signal then goes into the transverse tubule and then goes all the way through the muscle cell, through the transverse tubules, so that all of the myofibrils within the cell are exposed to the electrical signal nearly instantaneously. And that's all we have for this lecture recording. We focused on the structure of muscle cells. As always, if you have any questions, please feel free to shoot me an email or post them on the class discussion board. Happy studies.